So first I want to thank the organizers for the opportunity to make this presentation. So I'm going to tell you today about my work developing methods to help identify pathways that when mutated cause or drive cancer. And so first I want to give you some background on cancer and cancer research. So cancer is a disease that results due to the accumulation of somatic mutations during the lifetime of an individual. And a key subset of these mutations, so-called driver mutations, are responsible for cancer. And so one of the large and important challenges in cancer genomics is distinguishing the driver mutations from so-called passenger mutations, or mutations with no consequence for cancer. And so this is a challenge in large part because in a typical tumor, you may have at most 10, you generally have at most 10 driver mutations, while there can be hundreds or thousands of passenger mutations. And so in this diagram, I'm showing this model, the clonal theory of cancer in action, where you have a founder cell which at some point acquires a driver mutation, and as it accumulates more driver mutations, the replication rate increases and it becomes and grows into a tumor. And so in order to distinguish the driver mutations from the passenger mutations, we can sequence the genome at some point and use algorithms to identify the variants in the tumor genome compared to the healthy tissue. And so in this talk, I'm only going to be concerned with two types of variants, single nucleotide variants and copy number variants, and in particular, deletions and amplifications. And so to identify the driver mutations, we can compare the variation in tumor genomes across multiple tumors, for example, at the level of individual genes. And so if you've never looked at this problem before, your first thought is to probably look for the genes that have the most mutations or the most significant mutations. And so this is in fact what many researchers have spent time doing, and they developed sophisticated statistical significance scores where for a given gene you can weight the mutations by, say, their gene length, mutation context, or other attributes. And so researchers have applied these scores to large tumor data sets where if you were to plot kind of the results in terms of the significance score against each of the genes, you'd end up with a plot that looks something like this. And so here you have a relatively small portion of genes that are significantly mutated and a long tail of rarely mutated genes. And so this is interesting because we actually know that some of the genes or mutations in the long tail are important for cancer. And so here I'm showing a four-way Venn diagram where we compare three algorithms for identifying significantly mutated genes, MUT6TV, MUSIC, and OncoDrive, on the pan-cancer Cancer Genome Atlas dataset, which includes greater than 3,000 tumor samples of 12 cancer types. And so in the bottom left, I'm also showing, including a gene set from the Vogelstein et al. Review of cancer genomics from last year. And so as you can see in the middle, there are only 50 genes agreed upon by all methods, while there are hundreds found by only one method. And so this is because the long tail complicates the ability of these algorithms to identify all the driver mutations. And one of the explanations for why mutations in the long tail are drivers is that mutations target functions in cancer, or pathways. And so if you had two genes, A and B, where A and B interact to perform a particular function, then it's possible that if A is mutated more than B, and you only need to mutate A or B, that B could still be important for cancer, but wouldn't have enough mutations to be significantly mutated. And so in fact, it's very common to view cancer in terms of the pathways that are mutated in it. And so here I'm showing two examples of figures from TCGA ovarian and TCGA colorectal publications, where researchers have manually put together sets of genes that are in the same biological pathway that harbor many mutations. And as you can see, for these pathways, something like between 50 and up to 70% or even 80% of samples have mutations in these sets of genes. However, these figures were put together manually and have no notion of statistical significance. And so this is an important point, that it's still challenging to identify the significantly mutated pathways from a cohort of tumor sequences. And so one method people have used is to look at known gene sets or pathways and test them for enrichment of mutations. However, this misses out on some desirable features, namely novel pathways, crosstalk between pathways, and pathways that are active only in certain cancer types. And so researchers have developed methods to get around this problem by using knowledge of the somatic mutational process. And so I'm going to describe two principles you can use that to identify mutated cancer pathways. So the first comes from the observation that if in each sample you have few driver mutations and they're distributed across multiple pathways or functions, then there's approximately one driver mutation per pathway per patient. And so this gives us the principle of exclusivity. And so shown in this figure, I'm showing from Thomas et al., they did directed sequencing of a famous signaling pathway. And here the rows are genes, the columns are patients, and the red and black marks are indicating whether a gene is mutated in a particular patient. And so as you can see, the genes in the pathway have largely exclusive mutations. In addition, you'll notice that the genes, most patients have a mutation in at least one of the genes, which gives us the second principle, high coverage, meaning that many patients will have a mutation in an important cancer pathway. And so in 2012, Bandon et al. Developed an algorithm called de novo driver exclusivity, or Dendrix, where the goal of the algorithm is to find sets M of genes that display both high coverage and approximate exclusivity. And so the input to their algorithm would be something like this matrix shown on the right. It's a binary mutation matrix where each gene is either mutated in a patient or it's not. And so I'm showing it outlined in red, the highest weight set, where weight is the coverage minus the overlap in this matrix. And so the overlap is indicated by the orange bars, and the coverage is actually equal to the width of the matrix in this case, as every patient has at least one mutation. And so Van et al. Showed that finding the optimal set for an arbitrary matrix is NP hard, and they developed an MCMC algorithm to sample sets in proportion to their weight and show that the sets they could identify overlapped known cancer pathways. And one of the issues with their method, however, is that it's for identifying a single pathway, while we know that multiple mutations or mutations in multiple pathways are required in cancer. And so they discussed a method, which we have called Dendrix, for identifying multiple pathways greedily. So in particular, given a mutation matrix, you find the highest weight set, remove those genes, find the next highest weight set, and repeat to find multiple pathways. However, this method has some drawbacks because greedy can fail. In particular, because Dendrix doesn't know that it's looking for multiple pathways, it will group the most frequently mutated genes together, even if they're not exclusively mutated. So in this example, in this matrix, the highest weight set in this matrix consists of the two most frequently mutated genes and another relatively low frequency gene. However, if you're looking for the two sets with the highest total weight, you would pick these two. And so this observation led us to develop an algorithm called Multi-Dendrix, which we published in 2013. And so Multi-Dendrix identifies multiple pathways simultaneously. We formulated Multi-Dendrix as an integer linear program that can rapidly find the optimal solution for a given matrix and search a wide range of parameters to find stable collections of gene sets. And so Multi-Dendrix also has two additional features that Dendrix doesn't have, in addition to being able to find multiple pathways simultaneously. So the first is that Multi-Dendrix uses a parameterized weight function, W sub alpha, where I told you before the weight is equal to the coverage minus the overlap. Here, this weight function allows us to change the trade-off between coverage and overlap by multiplying the overlap by this constant alpha, or this parameter alpha. And so if you increase alpha, you will find gene sets with more exclusivity. In addition, Multi-Dendrix can search for gene sets of different sizes simultaneously. So it's searching for multiple pathways across a range of sizes simultaneously, while Dendrix is limited to a single pathway of a single size. And so now I'm going to outline the remainder of the talk. So first, I'm going to show how Multi-Dendrix outperforms previous methods on simulated data, both in terms of speed and accuracy. Then I'll show the results of applying Multi-Dendrix to mutation data sets from the TCGA, where Multi-Dendrix identifies gene sets that overlap known pathways. And finally, I'll discuss ongoing work extending Multi-Dendrix to large data sets and overlapping pathways. And so in order to determine if the innovation of the Multi-Dendrix algorithm actually improved its ability to identify cancer pathways, we used simulated data and compared Multi-Dendrix to the iterative version of Dendrix and the iterative version of another algorithm for identifying pathways de novo called RME. And so I'm going to quickly describe how we produce simulated data. The details are in the paper, but the basic idea is we have a mutation matrix with M genes and N patients, and we pick some of these genes to be planted or implanted pathways. And so we give them mutations that are both high coverage and mutually exclusive. And so then we mutate each gene in each patient uniformly at random, in other words, adding some noise. And so the idea of this simulation isn't to model all the complications of tumor biology or how pathways work, but to give kind of a simple case that we can benchmark the algorithms on. And so we produced 100 of these data sets for different noise rates. And for the 6 noisiest data sets, Multi-Dendrix outperformed the other methods in terms of average symmetric distance with p less than 0.01. In addition, we compared the run times of these methods, and we found that Multi-Dendrix is significantly faster on the simulated data. So in particular, it's kind of hard to see in this plot, but Multi-Dendrix is the bottom in each of these columns, the bottom bar. And that's because Multi-Dendrix completes in fewer than 5 seconds on each of these data sets, while for a subset of the data sets, it's orders of magnitude faster than the other methods. So you can't pick out how fast it is, but it's generally under 5 seconds. And so when we published Multi-Dendrix, we published it as part of a pipeline for identifying mutated cancer pathways. So we didn't want to just have the output and then have potential users have to figure out how to analyze and annotate it. So I'm briefly going to describe the pipeline. I don't have time to go into the details of all the methods. But the input to the pipeline comes from the mutation. So it's copy number variants, single nucleotide variants, and possibly other mutations that are filtered and combined into a binary mutation matrix. We then run Multi-Dendrix on this mutation matrix, and we perform 4 different types of analysis. The first is we analyze the results for subtype-specific mutations. And so mutual exclusivity and mutations can come from 2 genes being preferentially mutated in one cancer type or another rather than being in the same pathway. And so this is an important test to check for the results. We also have a permutation test to evaluate the significance of the results. And another way we evaluate the significance is using a direct interactions test, where we measure the surprise at observing a certain number of protein interactions among the set of genes in the output. And so the output of this pipeline is a collection of consensus modules, which are summarized gene sets from across different parameters, where we run Multi-Dendrix across a reasonable range of parameters in terms of the gene size and the number of pathways, and identify the stable modules of genes that appear together multiple times. So now that I've discussed how Multi-Dendrix performs on simulated data, I'm going to present results on real data. So we ran Multi-Dendrix on the TCGA glioblastoma data set that includes 398 genes and 261 patients. And so on the left of this plot, I'm showing you the stable modules identified by Multi-Dendrix, as well as their interactions. I'm color coding each of the modules by the cancer signaling pathway that they're found in, if they are found in one, which you can see in the legend in the lower right corner. And so one other thing that I'm showing is the mutation matrix, or the rows of the mutation matrix for each of these sets. And as you can see, blue indicates exclusive mutations. So these are all displaying that property, mutual exclusivity or proximal exclusivity. In addition, these gene sets are mutated in between 46 and 88% of the glioblastoma patients. And so one thing I really want to draw your attention to is that if you go back and look at the TCGA GBM paper and look at the signaling pathways they identified, they overlap pretty well with our sets. So in particular, our first set includes the 3 most mutated genes in the RB signaling pathway. The second set includes 4 of the most mutated genes in the PI3K signaling pathway. And our third set includes 3 of the 4 genes in the P53 signaling pathway. And so we actually can't even do better on the P53 signaling pathway, since CDKN2A appears both in the RB signaling, where we found it, and in P53. And so I just want to emphasize, we found these gene sets on the left with no prior knowledge of interactions, pathways, or genetic function. So we also applied Multi-Dendrix to the TCGA breast cancer data set, which includes about the same number of genes and twice as many patients. In addition to being twice the size in terms of patients, the data set includes more high coverage genes. So as a result, we increase this parameter alpha to find gene sets with more exclusivity. And so again, on the left, I'm showing the consensus modules identified by Multi-Dendrix, and I'm coloring them by the pathways in which they are found. And as you can see, many of them overlap known pathways. And in particular, the top left gene set includes genes from the PI3K and AKT signaling pathways and includes 6 interactions. So it's no surprise that this collection has a significant p-value in terms of enrichment for interactions. And so I'm going to begin to wrap up the talk by discussing ongoing work extending multi-dendrix to large data sets and overlapping pathways. And so our first extension was a new algorithm for multi-dendrix called that we call Multi-Dendrix MCMC. It's a Markov chain Monte Carlo algorithm where the goal is to sample collections of gene sets in proportion to their weight. And so the output of this algorithm is a distribution of solutions, including both optimal and suboptimal solutions. And so down here in the bottom left, I'm showing the example output where you have different gene sets or collections of gene sets, their weight, and a sampling frequency. And so this has already been released. We have the newest version on GitHub. And I just want to emphasize as well that its requirements are all open source. So you can download it, take it for a spin on your data set with no additional installation needed. And so in developing this algorithm, we noticed that there's an interesting way to summarize the relationship of the mutations between pairs of genes using the MCMC algorithm. So in particular, you can calculate the marginal probability of observing a pair of genes in the same gene set in terms of the output of the MCMC algorithm. And so we decided to construct a marginal probability graph where nodes are genes and edges connect genes and are weighted by the number of times you've found a pair of genes in the same gene set. And so this is similar to the multi-dendrix consensus approach, but it has 2 additional features that are valuable. The first is that you can find gene sets with unconstrained size and number. And so if you were to extract the high weight subgraphs of this marginal probability graph as your output, you would get this property. In addition, the gene sets can overlap. So if you find pairs of genes or sets of genes that appear sometimes with one set and sometimes with another, that might indicate that the pathways are overlapping. And so we have another dendrix, Dendrix++, that we're developing. And it also uses a new weight function I don't have time to describe today, but I want to give you a flavor for the results of Dendrix++ on real data. And so going back to that glioblastoma data set, we ran the multi-dendrix MCMC algorithm, Dendrix++ algorithm, and this is the marginal probability graph, or one of the high-scoring subgraphs in the marginal probability graph. And you can see that CDKN2A is kind of a bridge node between CDK4 and RB1 and P53 and MDM2. And in the bottom, I'm showing the mutation matrices again, where you can see they have relatively exclusive mutations. And so if you go back again and look at those figures in the paper, you see that half of this corresponds to P53 signaling, which includes 3 of these genes, and the other half corresponds to RB signaling. And so we're able to identify these overlapping pathways solely from the pattern of their mutations. And so just as a sanity check, Dendrix++ also identifies this portion of the PI3K signaling pathway as multi-dendrix had. So this brings me to the end of my talk. In summary, I presented multi-dendrix, a fast, exact ILP for identifying collections of gene sets with exclusive mutations. I showed how it identifies modules that overlap multiple cancer pathways in glioblastoma and breast cancer. And I presented Dendrix++, a new algorithm that can identify more complex pathways. And of course, none of this is possible without my research group or our funding and support. In particular, I want to acknowledge my advisor, Ben Raphael, and our collaborators on the Multi-Dendrix project, Roded Sharon and Dima Bloch. And thank you all for your attention.
Thank you very much. We have 5 minutes for questions, so lots of time for questions people have.
So thank you very much. That's a really exciting talk. I have 2 questions. The first question is regarding the input data size. So how many genes and how many samples do you think are suggested to get reasonable good results?
So in the examples I showed, we had about 400 genes and between 300 and 500 samples. And so I think you can scale up in terms of the algorithm's performance to more samples. But I think the key is in the type of cancer you're looking at. So some cancers have more mutations, which would take the ILP longer to compute on.
So basically, we need maybe more than 100 samples at least to start with?
No. I mean, in terms of a single cancer, it might have more mutations than, say, like leukemia compared to colorectal cancer. So it'll take the ability of the algorithm to scale depends on the number of mutations in the data set.
I see. Thank you. And the second question is for the output for Dendrix. The output is a list of significantly mutated pathways. Is it also possible to output how similar it is to known pathways? So we have an overview of the results about known cancer pathways.
Yeah, so this is definitely something we considered. And so we looked at actually the signaling pathways from the papers. Because they're actually a lot smaller, so the output of Dendrix and Multi-Dendrix is relatively small, so 3 to 5 genes generally. And most of the kind of known cancer pathways, say in KEGG, include many, many more genes, so it's hard to test them for enrichment.
Does it also output it, though? Does it tell?
Let's give some other people time to ask questions.
Sorry.
Thanks.
Yeah, so you were able to validate through Dendrix, or rediscover through Dendrix the hand-curated annotated pathways from the glioblastoma and breast cancer papers, but were you able to discover anything radically new?
Yeah, that's a great question. So I didn't have time to talk about it in the talk, but in terms of both data sets, we found genes that were interesting in terms of literature support for a role in glioblastoma and breast cancer. So I would look at the paper. But yeah, I mean, we like to think that because we're able to identify the known stuff, that the other discoveries we're also making are worth following up on. Thank you.
Looks like great work. Following up on that last question, this is de novo, so it doesn't know whether a gene is a tumor suppressor or an oncogene up front. Can the results tell us whether a gene is an oncogene or a tumor suppressor?
So in terms of the input is a binary mutation matrix, so in that sense, you don't have that information, but you could go back and annotate whether, say, the gene is, if it has copy number variants, amplified or deleted, or if there's a cluster of mutations in the gene. But it would be something you would do as an annotation at the end of the output.
All right, so this would just help us prioritize what we should look more closely at.
Yeah.
All right, thank you.
Thanks.
Thanks. Have you looked at how levels of alpha or the exclusivity changes between different cancer types?
No, we haven't gotten a chance to do something like that, but that would be an interesting way to do it. As I said, we've been working on this new weight function, which I don't have time to describe, but that kind of eliminates the need for alpha, so hopefully we'll see that soon.
Impressive talk. One more question. When you're identifying the multiple pathways simultaneously, is there a requirement of these pathways to be mutated in the same set of samples? I mean, when you calculate the coverage, for example, for one sample, is the coverage of the sample in different pathways to be calculated simultaneously, or they will be considered separately?
So you're calculating coverage across a collection of samples.
I see.
And so you're considering the number of mutations in that sample in the particular gene set, so you are considering them simultaneously. Does that answer your question?
I'm wondering, so you are maximizing the sum of coverage of different pathways from different samples. And this sample is being calculated in different pathways simultaneously or separately?
Simultaneously.
Okay, thank you.
Yes.
Great. And I have one more question. So the CDKN2B and N2A genes are head-to-head.
Yes.
Actually, no, they're tail-to-head. And they're deleted differently in different cancers. And I didn't quite catch it from your slide because you were showing mostly CDKN2A. I'm wondering to what extent the signal that you're processing for A is also signal for B.
Yeah, so we use the GISTIC output in terms of identifying the targets of the copy number variants. And so, while I neglected to mention it, the genes are actually both CDKN2A and CDKN2B grouped together, since it's kind of hard to distinguish those two signals.
Yeah, it's really interesting because in glioblastoma there's also microRNAs that are up and down regulated differentially, and some of those target the CDKN2A and N2B. So there's additional regulation. It'd be really interesting to see if you added microRNA deletion or expansion to the data. I don't know if TCGA even has it in the CNV data.
Mm-hmm. Yeah, I mean, that would be interesting, and since it takes in an input, a binary mutation matrix, you could easily add different mutation types or different.
Great. Great. Okay, well, let's thank the speaker one more time, and we'll go on to the next talk.